Welcome students, we are here again with another topic of your chapter cell, structure and function, and the specific topic is mitosis, which is also known as the equational division. In our last video, we have learned about the cell cycle and the various subparts of the cell cycle. And we know that in the cell cycle, there is an interphase and a divisional phase. The interphase has three substages, the G1, S and the G2, which is followed by the divisional phase. And the divisional phase is there where the cell actually divides into 2 or 4 cells depending upon whether it is undergoing a mitotic division or a meiotic division. Now, in this presentation, we shall learn about the mitotic division, which is also known as the equational division. Now, to give an overview of the historical perspective, mitosis was first observed by Strasburger in plant cells in 1870 and later by Fleming and Boveri in animal cells. However, the term mitosis was used by Alexander Fleming in 1882 and it is known as an equational division because a cell, a mother cell, we call it so, divides into 2 daughter cells, and the 2 daughter cells are not only identical to each other, they are identical to the mother cell, both qualitatively and quantitatively. Now, it is the somatic cells, the cells of the body, which undergo the mitotic division. Now, do all cells of our body undergo mitotic division? No. The germinal cells do not undergo mitotic division, but majority of the body cells, only barring the germinal cells, undergo this mitotic or equational division. You can see that the ova and the sperm, that means the male gamete and the female gamete forming cells, which are germinal in nature, be it in plants or animals, male gametes and female gametes are being formed by the germinal cells and they do not undergo mitosis, they undergo meiosis. But then that is a different topic, we shall discuss it in a different presentation. Now, why mitosis is also known as equational division? As I have already told you, because it produces daughter cells qualitatively and quantitatively identical to each other and also identical to the mother cell or from the cell from which it was derived. Now, you might remember that in the interphase, in the cell cycle, we have something called the divisional phase. Now, the divisional phase is also known as the M phase. Now, this M does not necessarily mean mitosis, it could be meiosis as well. That's why the term divisional phase is used there in the cell cycle. But in today's presentation, we are focusing on the mitotic cell division of the divisional phase. Of course, it is a continuous process, but for our convenience, we have divided it into 4 strict and clearly understandable parts, namely the prophase, the metaphase, the anaphase and the telophase. Well, what really happens before mitosis or for the metameiosis? Now, when I say what really happens, many things happens, but that we have learned in our cell cycle. But the most fundamental thing to remember is that before the cell or any cell for that matter could divide, of everything else, it must double its chromatin number or it must double its chromatin value. We know that each cell is twice N and 2C. That means it has a diploid chromosome number with a diploid state of its chromatin material. But when a cell is preparing for division, then in the synthetic phase, the S phase of interphase, it has actually doubled its chromatin, thereby the 2C status of the cell changes to 4C. Now the cell is ready with other components as well to enter the divisional phase, and here the mitotic phase. Now we shall go phase wise and look into the different things that occur in each of the phases. So the first phase is prophase. The prophase is a bit longish, and if we want to, we can have further substages as early prophase, mid prophase, and late prophase. Now all these are stages that we give ourselves depending on the activities the cell is doing. We see that in the early prophase, the chromatin material within the nucleus, which is otherwise not visible, becomes thickened and start to condense. They seem to form thread-like structures, a clump of threads, if you may call it that way. Then by the mid prophase, they are further thickened and shortened, and we see that the centrosome has started to divide to form two centrioles on the upper pole of the nucleus, the outside of the nucleus. By late prophase, the nuclear membrane and the nucleolus begin to disintegrate, and the two centrioles so formed on the upper end pole start moving towards the opposite pole in the equatorial direction. Now that gives an impression that the entire cell is trying to reorganize and do something which is different from its normal activity of physiological function. We also observe that in between and from the centrioles, certain thread-like structures or astral fibers are coming out. They are called astral fibers because they look like stars when they come out from the centrioles. Now these fibers are actually the microtubules which form the spindle fiber and are made up of tubulin protein. The cell at the end of prophase do not show the Golgi complex, the endoplasmic reticulum, nucleolus, nuclear envelope. That means these organelles also disintegrate and get diluted in the matrix. Now we can see the picture that in the prophase, the cell is gradually preparing for division, and by late prophase, the nuclear membrane have started to disintegrate, and also you can see that the two centrioles are separating. It was a single structure, the centrosome. The two centrioles are moving away, and you have a fluorescent micrograph where you can see the an actual cell which is into the prophase stage. Now we can go into the metaphase, which is the phase immediately after the prophase, but there is no gap as such. There is no stopping and beginning. It is a continuum. There is a complete disintegration of the nuclear membrane and nucleolus. The chromatin fibers further condenses, and we see that the chromatin fibers break into pieces, and now they are more visible as chromosomes. The chromosomes have their centromere or the kinetochore, and we see that these chromosomes start moving towards the equatorial plane and begin to align themselves in a vertical line. The spindle fibers form further as the centrioles go away to the opposite poles. The spindle fibers come from the opposite poles, and one spindle fiber from either end would attach a particular chromosome at its centromere. It is something like a rope is being tied to the waist of a person who is standing with his hands and legs apart. So that is the situation in which all the chromosomes are aligning on the vertical plane in the middle of the spindle fiber, which is horizontal. You can see here that gradually the centrioles have moved apart, and the chromosomes, which are actually now showing a double status, because if you remember that the chromatin had already doubled, so each of the chromosomes are actually showing a tetravalent structure. Okay, so they have got 4 chromatids and 2 chromanimata, and there is one kinetochore. That means each one is like an X, but the arms of the X are still not spread out. So in this way, here you can see there are 4 chromosomes in the picture, but for the human cell there would have been 46 chromosomes, and each one doubled in its entity. That means in actuality there are 46, along with them another 46, so there is actually 92, but we are looking at them as 46 in pairs. So we can come to the next phase, which is the anaphase. Now this is the most important phase for the mitotic cell division. During anaphase, the spindle fiber starts to contract. As the spindle fiber starts to contract towards the opposite pole, they exert a pull on the centromere of that chromosome, that X-shaped chromosome, and try to tear the 2 partners from that one jointed area, which is the centromere, the waist, if you may call it. So the X, which is standing with its 2 hands up and 2 legs down, and the center is attached, the thread from either side will pull away the center, so that on each side you have 1 chromosome. Remember, the chromosome had already doubled earlier. So what is happening now? The doubling is only becoming separated, but by no means it has become qualitatively half or quantitatively half. It is the double of the sum which is coming down to its normal value. So each chromosome moves away from the equatorial plate. The centromere of each chromosome is towards the pole, and hence at the leading edge the arms of the chromosome trail behind, and they make this L, V or J-shaped structure, because tie a rope to a waist, and if you pull it, then the whatever person or whatever it is, the legs and hands will go on the forward direction. Depending on the position of the centromere, whether it is exactly at the center or whether it is off center, the structure of the chromosome would be either V-shaped or L-shaped or J-shaped. Okay, so in anaphase, this very situation or the visibility of chromosomes in VL shapes prove or show us that the chromosomes are being pulled away from their center towards the opposite poles by the spindle fibers. Here you can see pictorially how the pairs are now being separated and are being pulled away. So you have some of them forming L shape, some of them forming V shape, because some chromosomes are metacentric, some are submetacentric. There might also be telocentric chromosome. In that case it will be I-shaped, and you can see in the fluorescent micrograph picture how beautifully it is clearly shown that the chromosomes are being pulled away by the green colored. Here the color green has been used to show the spindle fibers, and blue depicts the chromosomes, the pairs of chromosomes which are now have been separated and been taken to the separate poles. The last phase is the telophase. Well, by now the separated chromosomes, if you consider the human cell, then 46 on one end and another 46 on another end. Remember, within the same cell, the chromosomes have reached their 2 poles, and around those chromosomes, new nuclear membrane starts forming. New nucleolus within each of those nucleus appear, or you can say reappear. The organelles also start reappearing, and by late telophase, there is something like a constriction at the center of the mother cell. So it forms a dumbbell-like structure with two bulges on the sides, with two nucleus on two sides, and within those nucleus 23 pairs, that means 46 chromosomes, and 23 pairs 46 chromosomes on two sides. We had started with 46 chromosome before the interphase. In the interphase, the cell has doubled its chromatin value, and then by mitosis, it has again regained its twice n value from 92. That is, it has become 46 from 92. That means the chromatin value when it was 4C. So the chromosome cluster, the opposite spindle poles, their identity is lost as discrete elements. Nuclear envelope and all those components I told come back, and the nucleolus is also there, Golgi body is also there. By that time, the spindle fibers disappear, and the two centrioles now act as centrosome for the respective would-be cells. But remember, even now, the center of the cell is constricting. Now, if it is an animal cell, it will constrict, and the constriction will be brought about by annular formation of microtubules, which will gradually constrict the cell and cut off the mother cell into two. But if it is a plant cell, then there will be Golgi bodies, which are known as phragmoplasts, which are packets of substances which will come and lie at the center of the cell, and they will form a wall. A new cell wall will be created at the center of the mother cell, thereby one cell will become two cells. In case of plant cell, there is no constriction because there is a cell wall. A new cell wall is formed. Now that here you can see in telophase there is constriction. So in the electronic micrograph also you can see that there is a clear constriction shown, and this is what happens. The new nuclear membrane, which is shown in yellow, has come. The centriole is now being converted into centrosome, and the cell is undergoing constriction. Now, these four stages, the prophase, metaphase, anaphase, and telophase, marks the end of the chromosomal reorganization or chromosomal separation, and they are taken away to the opposite poles. And that is followed by the physical division of the cell, as I said, and that is what is known as cytokinesis. Cytokinesis, as I have just mentioned, occurs in these two ways: by the constriction of the mother cell by the microtubule formation at the center, that's for animal cells, and by the formation of a new cell wall through phragmoplasts in plant cells. Here you can see that the animal cell or one single cell is divided into two cells. A real picture is there, and of course for a plant cell also there is a real picture where the forming phragmoplasts at the center is being shown in small bubbles. So those Golgi apparatus components will coalesce, will mix with one another, and will give rise to the cell wall. In the schematic diagram you can see how a plant cell divides into two plant cells depending on the formation of the cell wall. But on the other hand, for the animal cell, you can see that the mother cell is constricted, and the constriction, as I said, is because of the microtubular ring formation. The result is two daughter cells identical in every aspect, not only to themselves but also to the mother cell. Now, what is the significance of mitosis? I believe you've understood the process of mitosis, but then what is the significance of mitosis? Well, we can take three important words: growth, repair, renewal. Think of those days when you were small, you were a baby, and look at yourself in the mirror today, or look at your parents or your grandparents. The process of a tree, which is a seedling, and then growing into a huge banyan tree, or for that matter, somebody had a fracture and then its repair, or a cut and its repair. The blood cells, which are dying every 3 months, and their renewal. So all of this process of cell repair, cell renewal, growth and development, the dead cells from our skin, the nails, the hair which are growing, are being constantly replaced and renewal. Had there been no renewal and replacement, then like any other inanimate object, on friction our legs would have become reduced and would have come all the way to our waist maybe, because with friction the cells would have gone away. It is only because there is constant renewal of the cells that we maintain our selves. After a certain period of growth, never think that renewal and repair has stopped. It will not stop till the person or the organism dies. But then we see the visible change when it is young and then becomes adult. But after adult, maintenance is there, replacement is there. So the main significance of mitosis is the maintenance of the biological system, the growth of the biological system, and the renewal of those cells which have a periodicity and have to be replaced by fresh and vigorous cells. I hope you have understood how and why this process occurs, why mitosis is known as an equational division, and what are the stages that we can divide it into for understanding clearly the processes involved in the division of a single somatic cell into two cells, however very much identical in every respect. Their functionality, their components, their qualitative nature, everything is very much similar, and that is what maintains our life process. Well, just think, the picture tells it all. The baby elephant and the mother elephant, what a huge difference in size. Yes, it is the magic of mitosis. Think over it. Thank you.